The paper I'm going to be presenting for A&P is Circular and Longitudinal Muscle Shortening indicates sliding patterns during peristalsis and transient lower esophageal sphincter relaxation by Nirali Patel et al. For a little bit of background, let's first start with the upper digestive tract, especially the esophagus. From the throat, food enters the esophagus, a hollow muscular tube about 10 inches long. When you swallow, the upper esophageal sphincter relaxes to allow the food to enter the esophagus. After you swallow, the sphincter contracts to prevent air in the throat from entering the esophagus. The epiglottis closes over the trachea to prevent the food from entering the airway or the lungs. So the esophageal movement can be bidirectional. First, we have peristalsis, where a series of strong wave-like contractions in smooth muscle carries food from the esophagus to the stomach. This is known as aboral movement. We then have transient lower esophageal sphincter relaxation, or known as TLESR. This is a major mechanism of acid reflux, where contractions carry contents of the stomach back up through the esophagus. This is known as oral movement. Now let's look at the layers of the esophagus. The two major layers that we want to know for this paper are the muscle layers. First is the outer longitudinal layer, known as the LM, which contributes to the actual shortening. Then there's the inner circular layer, or the CM, which contributes to axial shortening. This muscle layer has a spiral morphology and is known to increase luminal pressure. Now let's take an in-depth look at peristalsis. First, smooth muscle tissue, both circular muscle and longitudinal muscle, contract in sequence to produce a peristaltic wave, which propels a bolus along the digestive tract. The peristaltic movement comprises of relaxation of circular smooth muscles. A contraction behind the chewed material keeps the bolus from moving backward. Then, a longitudinal contraction helps push it forward. Now let's look at transient lower esophageal sphincter relaxation. TLESR is the major mechanism for acid reflux, where contractions carry contents of the stomach back up the esophagus. Longitudinal muscle contractions are observed near the end of the esophagus, near the stomach, but circular muscle are not observed at all. So the main point of this introduction is to show that sliding motion between the layers of longitudinal muscle and circular muscle is seen when different relative shortenings or contractions occur between the two. So the purpose of this paper was to study the contraction patterns of circular muscle and longitudinal muscle during peristalsis and TLESR. Now let's look at the materials and methods. For this study, both human and rat subjects were used. However, for this presentation, we will only be looking at the human results. 18 human subjects were used. They range between 20 and 44 years old with no history of gastrointestinal disease or surgery. Subjects had to fast and stop smoking 6 hours beforehand. Subjects' esophagus were locally anesthetized in order to minimize discomfort of the pressure-sensing catheter they must swallow. Subjects were allowed to swallow water in 30-second intervals 5 times. Pressure readings were taken in 2 locations, 6 centimeters from the top of the esophagus and 12 centimeters from the top of the esophagus. Scientists concluded muscle thickness from pressure readings from the pressure sensing catheter. Now let's look at some of the results. In Figure B, we can see the summary of findings from a group of muscle contractions. SW stands for swallow, which is the indicator for peristalsis. TLESR indicates itself. CM and LM still stand for circular muscle and longitudinal muscle. We can see the max thickness compared to the baseline thickness of smooth muscle during a muscle contraction. Baseline or normal muscle thickness is known to be 1. Circular muscle seems to triple in thickness during peristalsis, while longitudinal muscle also seems to increase slightly. Circular muscle thickness has no change during TLESR, but longitudinal muscle thickness seems to double. The third data set is the comparison of both longitudinal muscle and circular muscle to max muscle thickness. The error bars indicate standard error from the average peak. For this figure, we will look at muscle thickness during peristalsis and TLESR. For the baseline model, longitudinal muscle and circular muscle are the same thickness because esophageal muscles are at rest. For the swallow model, or the peristalsis model, circular muscle is much thicker and shorter than longitudinal muscle, but compared to the baseline model, both muscles are thicker and shorter than the baseline. In the TLESR model, circular muscle appears to be the same thickness as baseline, while longitudinal muscle seems to be much shorter and thicker than the baseline. From these figures, we can conclude that both the circular muscle and longitudinal muscle within the esophagus move independently of each other. The muscle contractions of peristalsis and TLESR have a direct effect on the thickness and shortening of the muscle layers. These are my works cited. I cite the article and my figures that I used. These are my 2 class discussion questions. Why is it important for CM and LM layers within the esophagus to have a sliding independent relationship to one another? Also, why do you think that circular muscle plays a large role in peristalsis but no role in TLESR?